Excellent. So our next talk was by Will Koval.
Great. Thanks everyone for coming. Really appreciate being here. Persistence is going to sneak up on us a little bit rather than Emily's great talk. I'm going to be talking today about capturing spatial uncertainty. And the uncertainty that I'm talking about here is the explicit random variation that we incorporate into our models in order to alter infection dynamics. That random variation, or stochasticity, I'm sure I don't need to tell all of you, comes in a couple different forms. One strategy that we can use to incorporate stochasticity into our models is environmental stochasticity. This is kind of parameter noise that we include to approximate some environmental process that we don't explicitly include or even know about in our epidemic process. And these random events that befall populations change the infection dynamics over the course of infection. Contrast that with demographic stochasticity, which are the random events that befall individuals. We're tracking each individual's life history. There's a stochastic ordering of the events of transmission, decay of pathogen, et cetera. And that changes the epidemic outcome based on the order of those events. Because it's individually based, larger populations seem to approach deterministic cases. And sort of to dive a little bit deeper into that, we know that as host density increases, epidemic size, or the cumulative fraction infected, increases. This is just a deterministic model without random variation. And if we change the absolute values of these parameters, but maintain the ratio, we see that we can just recover these same curves. The relationship between cumulative fraction infected and host population density with respect to the epidemic threshold is the same. When we account for environmental stochasticity, we get a little bit of noise around this cumulative fraction infected, but the relationship is constant across these three absolute sizes of the populations. Whereas with demographic stochasticity, we don't see that relationship. There's an actual correlation between the absolute value of host population size, where in smaller populations, we have higher variability and we have reduced probability of infection. And as we increase host population size, you can get the deterministic case, recover that with low variability as we approach the cumulative fraction infected. So these demographic stochasticity effects weaken with increasing population size, but what happens when these individuals aren't perfectly mixing and they're distributed over space? As Mike Boots was talking about yesterday, space, stochasticity, and demography interact in interesting ways to qualitatively alter mean model behavior. And what's really exciting now is that we have the computational power to actually fit spatial stochastic models, hopefully not ending up in this image. But we can fit the mean of these models to pooled epidemic data, but the spatial distribution of infection rates over time differs between the two models. So we really want to understand what is actually the spatial distribution look like and how are we capturing the mean dynamics later. So there's a couple of different advantages to each of these stochastic sources. Demographic stochasticity, of course, is a mechanistic way of interpreting transmission, whereas environmental stochasticity is this flexible dynamic that we can actually account for multiple environmental processes. So the question here is which stochastic source is actually informative for the spatial distribution of infection rates? And maybe a little bit more cynically, the question that we're all really trying to get at is how complex do our models need to be in order to explain our epidemic data? To get at those questions, I'm going to be talking about a story from the Douglas fir tussock moth, a system that we work in, which is a forest outbreak insect that's affected by a baculovirus, species specific and environmentally transmitted. The caterpillars pick up virions from their environment by feeding on leaves that are contaminated with the virus. They incubate these inside their cells, and eventually the host integument ruptures, leaving virions all over the leaves for other caterpillars to pick up. Just to give you an idea of the spatial scale of these populations, and the contrast on these photos maybe isn't that great in this light, but in the top panel, I'm showing you a not so good photo of several million caterpillars. And in the bottom panel, we have what they've done to the forest landscape. So the defoliation that we see in these photos would be worse without the virus, which makes us one of the few groups where infection is like a really good thing. And we are trying to operate at really large spatial scales, large population scales, in order to understand how this random variation affects the distribution of infection. We collect data from the system by getting ideas of how many caterpillars are exposed over time. We use an elegant system of me whacking a tree with a stick, and we get out some caterpillars from that, quarantine these individuals separately, and make sure that we get an idea of how many were exposed at the time of collection. So we can track local contact groups so that we can get an understanding of this implicit spatial epidemic data. And then we're going to fit multiple spatial models to this data for model comparison. We have several populations that we were monitoring, which range over host population sizes from an average 1 insect per branch to an average 150 insects per branch. So that's a large range. And again, we're including this implicit spatial data where we're keeping track of which caterpillars are directly interacting with whom for local contact groups. Sort of on the theme of persistence, I was supposed to go out and collect this data myself in summer of 2020, and that didn't happen. This project almost fell apart, and thanks to these amazing people, our collaborators at the Forest Service and Department of Natural Resources, we were actually able to get this project done thanks to their sampling efforts, which was really amazing. Moving on to the models. At the core, this is an SEIR model, and we're incorporating movement of susceptibles and exposed individuals. Cadavers don't move. And it's in explicit two-dimensional space. And crucially, we're accounting for host heterogeneity and susceptibility. More susceptible individuals are removed with higher probability. We're including demographic stochasticity through an agent-based model. So that's the stochastic ordering of events. And we're accounting for environmental stochasticity with random reaction diffusion models. That's a time-varying transmission coefficient. So there's like random fluctuations in how fast transmission is occurring. And then we're actually parameterizing these models with our spatial epidemic data using direct likelihood Bayesian inference MCMC. If you want to talk to me about that, feel free. I don't have time to get into it right now, but we're going to then use an information criterion to compare the explanatory power of these two sources of stochasticity. And crucially, because we have this range of host population sizes, we can get at the information criterion on the y-axis, relating that to insect density on the x-axis. And in the middle here, where we would have no informative difference between the models, in smaller populations, we may expect that demographic stochasticity would be more informative, offer more explanatory power, and environmental stochasticity would offer more explanatory power in larger populations, with possibly no difference in between, but we're not going to make any claims about that prior. Whoops. So our traditional approach in the lab is to just kind of take this model, project it forward, calculate the fraction infected within a simulation, and then we gauge the probability of our data given the mean fraction infected in that simulation. But this isn't the model structure that we have. Instead, we have these spatial models that have structure in them, that have variation in fraction infected over space. And in the left panel, you see the actual simulation, and in the right panel, the change in fraction infected over time. So we're calculating the probability of our data given the distribution of these fractions infected in order to parameterize the models. Now, when we have our spatial data in the top panel, where circle size is denoting the actual number of samples that had that fraction infected, and comparing them to multiple model realizations on the bottom panel, we're getting out a final posterior distribution of the fraction infected over time. In the top panel, I have demographic stochasticity in green, where darker colors denote higher posterior probability of observing a fraction infected at that time point, and in blue, in the bottom, environmental stochasticity. What I want you to notice here is that the environmental stochasticity is more diffuse. It has less sharp contrast between the high probability and low probability regions that we see in the demographic stochasticity model over time. And also importantly, there's these refugia that appear of virus-only or susceptible-only areas of really low or really high fraction infected in the demographic stochastic model. These features are accentuated in lower population densities, but still persist in higher population densities. We still get the same dynamics of refugia, which is pretty common in theoretical spatial models. But now we're parameterizing them with this spatial data set over time from several of our populations. So, like I said, stochasticity smooths over this mean. Demographic stochasticity makes this distribution sharper. And instead of going by I, we can actually get an information criterion out of this, saying that demographic stochasticity is more informative for our system. But what about this relationship of populations and the actual explanatory power that this possesses? We didn't see this relationship at all. Actually, in smaller populations and the largest populations, we had no informative difference between the two models. And we actually see an increase in the explanatory power of demographic stochasticity with population size. And these strengthening effects of demographic stochasticity's explanatory power run contrary to what we would expect from stochastic theory. Now, grounding ourselves one more time in what this means for epidemic size, the demographic stochasticity model has a lower predicted cumulative fraction infected. And so with this reduced infection, there's higher defoliation potential. And so these models actually have meaningful effects for real world situations, which we've seen in some of our study sites where we have high densities of insects and they fail to collapse due to low infection rates. So we have this empirical test showing strengthening effects of the explanatory power of demographic stochasticity, real world consequences observed, and we think that this will have meaningful consequences for future outbreaks of this moth. And also just to reiterate that we need to be careful about how we incorporate stochasticity in our models, be very purposeful about it. And we suggest that this pattern would likely hold for other environmentally transmitted diseases of wildlife. With that, I'd like to thank all the people who make this project actually fun to work on, and specifically give a shout out to the Forest Service, Department of Natural Resources, and other governmental institution folks who have made this a tremendous way to connect entomology, disease ecology. And with that, I will take any questions. Yeah.
Hi there. Yeah, that was a really interesting talk. If I understood you correctly, your environmental noise model is effectively stochastic differential equations with time-varying parameters. In my own work, I've played around with them quite a bit and noticed that they're pretty sensitive to how you scale the noise process.
Yeah.
Did you look into that when you were doing this model, and did that affect the results that you got?
Yeah, for sure. It's a great question. So we're talking about how we incorporate the stochasticity. Really, these are random differential equations, not stochastic. There's no exact continuous time noise. We're changing the coefficient of how we multiply, like a random draw. We exponentiate a normal distributed parameter and multiply that by the transmission rate. And so log normal distributions, obviously the expectation is going to be higher than 1 unless you make some specific adjustments. So depending on what distribution you're drawing from, yes, you're going to see probably higher cumulative fraction infected. But because we're parameterizing the variance of that noise within this model, we're getting penalizing for too much breadth and trying to get at the actual distribution. Yeah.
So I was wondering, could you reiterate what the pathology for the caterpillar is with this virus, and whether you think your sampling method could bias your collection of infected individuals given that pathology?
Yeah, it's really the perfect question, because we're sampling all in the lower canopy, mid canopy, what's accessible. These trees are 30 meters tall, depending on where you're sampling them. And the caterpillars are distributed in that 3-dimensional space. So what we're considering with this 2-dimensional model is that they're moving within the same canopy region, and they're not moving up 30 meters, but they're spreading out within trees that touch each other. So really, we're trying to build a model that captures where we're sampling.
Thanks for the great talk. Very, very interesting. So in addition to the different scaling properties that you point out between these different forms of stochasticity, they differ in the way that they handle the zeros, the local absences in your data.
Yeah.
And I wonder, to what extent was the increase in explanatory power that you found driven by just their ability, the demographic stochastics model ability to account for those zeros?
Yeah, so I was worried about this kind of like just denominator effect of like, we're sampling, there's some number of insects, but we also fit models where there's no movement. So basically bundling stochastic ODEs together. And those are able to have the zeros and ones within the distribution. And they actually performed worse worse compared to our reaction diffusion models. So there's some sites where, because they're not interacting with other random ODEs, they end up with very high infection or with basically zero just due to the random initial conditions. And so those models don't do well. It's really the demographic stochasticity playing out early and late in the epidemic, if that makes sense.
Hi, at the risk of making your computers collapse, do you need to incorporate y-axis at all in any of the spatial models? Like, does it matter where on the tree the infections are occurring?
If I understand the question correctly, you're talking about the way that we set up the grid within the simulation?
Yeah, thinking about sort of height.
Yeah.
So if you've got like infections occurring at the top of the tree, do sort of virions fall down to lower levels where they can infect insects lower?
Yeah, before the resonance cut you off, I think I understood your question. Yeah, so we know that in 2D and 3D, in theory, there's different invasion speeds of virus in these models. And we're, like I said earlier, because we're just operating within a similar level of the canopy where we're imagining that there's limited interaction between higher areas that are many meters away as opposed to local, we're pretty sure that we're able to capture at least some of the dynamics. Also, we have perfectly mixing models of instead of explicit space, just an implicit metapopulation model. And there's not a lot of meaningful difference in the parameters that we get out from there. Yeah.
Excellent. I think we need to thank Will and transition. So thanks very much.